Please subscribe Ramzan Biology for next lectures.
Assalamu alaikum, welcome back to Ramzan Biology. In this lecture we will talk about the life cycle of a cell. That what is a life cycle? Life cycle is actually the phases from which a cell passes during its completion, during its completing of its cycle in order to produce some data cell. So life cycle is actually the cycle from which a cell, a mature cell, passes and produce its daughter cell. Today our topic is about the life cycle of the cell. So if we talk about the life cycle of a cell, so life cycle of a cell is divided into two phases. One is the interphase. The first phase is called the interphase, second is called the mitosis. So a mature cell completes its cycle in two phases. One is the interphase, second is the mitosis. Interphase is actually the synthesis phase. It is a phase in which a cell prepares itself for the next phase. This is synthesis phase and this is called division phase. This is the phase in which cell divide and produce daughter cell, and this cell support and this phase support this division phase. So we will take this with example. So if you take this is a mature cell, this is a mature cell, this is the nucleus of this cell. So if you take this as a mature cell, so when it undergoes its life cycle, so it will undergo the two phases, interphase and mitosis phase. The first phase is called the interphase. In the interphase, this mature cell, this will produce some DNA, some protein. So this phase is called the G1 phase. It's called G1 phase. And the next, if we talk about so this cell will come there. Okay, so and it's preparing in the beginning of the interphase. The first phase is called the G1 phase. So interphase divided into G1 phase. In the G1 phase, we say that for division of the cell, for producing two daughter cell, a cell have to duplicate its genetic material in order to produce its daughter cell. So firstly, for duplicating, there is need of some supporter, some food. The food is protein and enzyme. So in G1 phase, this phase, the synthesis of protein and the enzyme occur. So we call it this phase is called the pre-synthetic phase. Okay, the next phase is the S phase. This is called S phase. In this phase, the duplicating or replicating of genetic material occur. Like if you take this as a DNA double helix structure, take more clear. This is a DNA, so it will unzip, and in between it will produce genetic material. So this is the parent DNA and it will produce the sister DNA. So the genetic material of the cell are replicated and the parent genetic material are produced. So the G1 phase produce protein and enzyme, and the S phase the synthesis of the duplicating of genetic material occur. Now the cell will enter another phase. This phase is called G2 phase. One called G1 phase gave one, this called G2 phase gave 2 phase. In this phase, synthesis, in this phase also some synthesis occur because the cell is now prepared to be divided, to be enter the mitosis phase. So for mitosis phase there is also need of some protein. So in the G2 phase the synthesis of protein also occur, protein synthesis. Okay, protein synthesis. Now the cell is ready to be enter into the mitosis phase. Okay, so this is a cell. Now in this cell the mitosis process start, in which we say that in this cell the motion of the genetic material occur to opposite pole of the cell, and then it undergoes the whole phases of the mitosis and eventually give us two cells. This also have nucleus, this also have nucleus, and have the genetic material of their own. After completing this cycle, some cell have the ability to again start this cycle, while there are some cell or tissue which are not able to again start this cycle, and they enter another phase which we call as a G0 phase. So after this enter another phase, this is called also resting phase, or we can also call as a G0 phase. So while when some tissues or cell enter to G0 phase, while some have the ability to again enter the interphase and mitosis phase. So on the basis of these three cells, there are three types of cell or tissue in our body. The first type we studied that some cell have the ability to again start the process of life cycle, while so these cells are called lobule cells, and many these cells are found in our gastrointestinal part. This is gastrointestinal part, and also in our skin. Okay, the skin. So the lobule cell which have the ability to directly enter again the life cycle, the interphase and the mitosis phase, these cells are called lobule cell. These are found in the skin and the gastrointestinal cell. These are the cells which need more cell to produce. So for more cell, it have to regulate this cycle again and again. So therefore, these are the cells which have the ability to circulate this life cycle again and again. Now, the second type of cells are the second type of cell are called stable cell. They are called stable cell. These are the cells which enter the G0 phase but have the ability to again enter the interphase. Okay, so these cells when enter from the producing two daughter cell into the G0 phase, so how it again enter to the interphase? It is therefore when the G0 phase cell get the stimulus from the body to produce and again enter the life cycle of the cell. So therefore this cell again enter to the interphase and mitosis phase, and these cells are found in our body in the liver, the kidney, so and the pancreas. So how these cells again enter in the interphase? Like if I take there this is a liver, so this is a liver. If I cut this part of the liver, so now the liver become half. So it have already entered into the G0 phase when the liver complete. So when I cut its little part, so now the liver cell which are called stable cell, they got a stimulus from the body that you have to again enter and circulate the interphase and mitosis pain in order to pull the liver. So therefore the liver cell, stable cell, hepatic cell, these are called, and the liver cell are called hepatic cell. So the liver cell again enter from the G0 phase into the interphase. So therefore it become full liver, and also the kidney also, in the pancreas also. The third type of cell are called the permanent cell. Permanent cell. Okay, as we know that the permanent will refer to that thing which is permanent. It is endless. It means that these are the cells which don't have the ability to again enter into the interphase from the G0 phase. So these cells are found in our body in the nerve cell. These are the nerve cell, and the second is the skeleton cells. So the nerve cell and the skeleton cell are the permanent cell which don't have the ability to again enter from the G0 phase into the interphase. So therefore the nerve cell and skeleton muscles are not able to elongate anymore. So in the conclusion of these three types of cell and the three types of motion after the mitosis process, we say that every cell in our body is under the stimulus of our body. It mean that the every cell when divide and goes interphase and mitosis, it receives some stimulus from our central nervous system to circulate this cycle. If there is no stimulus, so they cannot. If suppose there is no stimulus giving to the cell, a somatic cell of the body, circulating this cycle, so our every part of cell, our every cell of the body will circulate this cycle again and again, and our ear will elongate, our nose will elongate. So therefore, to control this motion, like small embryo have small nose, small ears, and all have small things, but the big men have big nose, big ear. So if we see to our nose, that our nose is smaller than our hand, so therefore it is under the control of some stimulus of our body to control the regulating of the life cycle of the cell. If there was no this stimulus, so our nose will elongate like our hand. So then the stimulus will be not, and we will not be like this as now I am looking this. Okay. So in the conclusion, we say that all the cell in our body are under the control of some stimulus from our central nervous system. Now up to this, we only studied about the general structure of the life cycle of the cell. Now we have to conclude about the phases of the mitosis, that in the interphase the synthesis of genetic material occur, what occur during the mitosis process, in which two daughter cells are produced. So we will talk about the mitosis. So mitosis is again divided into several phases. There are 4 phases of the mitosis. They are PMAT. I write here as a PMAT. P mean prophase, M mean metaphase, A mean anaphase, T mean telophase. So these are the phases of the mitosis. Okay. So now we are going to start with the prophase. So before, from the prophase, we will take, there is a cell. This is a mature cell. You can see here, this is a mature cell. It has the nucleus inside. So we take this as a mature cell. So before from the prophase, they have the genetic material, and the genetic material form is a network. Okay, so this cell is actually founded in the G2 phase. Okay, so from the G2 phase, when the cell have duplicated its genetic material and then enter into the G2 phase and then synthesis the protein, now the cell will directly enter into the mitosis, and the first part of the mitosis is the prophase. So before the prophase, the cell nature, the cell structure outstanding is like this. The genetic material are uncoiled and like this structure. So now we will start with the prophase. So in the prophase, this is a cell. Okay, so now this is a cell. In the prophase, some three very important events occur. Firstly, the genetic material, these are uncoiled and form a network. These become coil like structure, and they coil in the coil like structure are called the chromosome. These are called as the chromosome. And the second is each chromosome is with its copy. What is the copy? Copy is the second chromated part of the chromosome. Okay, so from where this copy occur is before in the synthesis phase, as we studied that in synthesis phase the genetic material DNA of the cell are duplicated with the help of protein and enzyme formed by the G1 phase. So the duplicated DNA, they produce some sister DNA. So the sister DNA then form a sister chromosome. So now a chromosome, this is called, if I take this is called one chromosome. When it is with its sister, with another chromosome, so now we call these are two sister chromatid. So now each chromosome is with its sister chromatid. The third very important basic structure talk is this, that this nuclear membrane which was in the G2 phase, it disappear. You can see here that it disappear, and now the chromosome are free to be flow in the cell. And before there were this centriole, beside the nucleus. So now in the prophase, the centriole move to different pole of the cell. There also and there also. It is a little in between this chromosome. So what occur in the prophase? Firstly, the genetic material uncoil or become coiled and become a chromosome like structure. Each chromosome is with a sister chromatid, and the nuclear membrane disappear, and the centriole move to different pole of the cell. So the prophase is completed. This phase is called prophase. Now the cell have to enter another phase they call metaphase. So this is a cell. When we take this cell, so these are the centriole from upward pole of the cell. So now in the metaphase, three types, the centriole produce some spindle fiber with the help of protein. As I mentioned before that in G2 phase the synthesis of protein occur, and these protein help in the production, the creation of spindle fiber and the metaphase of the mitosis. So now with the help of G2 formed protein, the centriole create spindle fiber which help in the motion of the chromosome during the metaphase and the second anaphase. So Now these centriole then produce three types of spindle fiber. As you can see, these small spindle fiber, as I mentioned, these are called estaural spindle fiber. These small are called estaural spindle fiber or estaural microtubule. The second are these are the spindle fiber which we call as a polar microtubule. These are called polar microtubule because they start from one pole of the cell and attach with another pole of the cell. A third type of the microtubule are the another type. Before I write as a chromosome, there also this is a chromosome and this is another chromosome. This is with its coffee. Okay. So now the third type of spindle fiber which will be formed by the centriole are the kinetochore spindle fiber. These are the specific spindle fiber which attaches with the centromere of the chromosome. And if I take there as it is a chromosome, this is a chromosome, so this point is called where the chromatid attaches with each other and form a whole chromosome. This part is called centromere and the each side is called kinetochore. So therefore the spindle fiber which attaches with the kinetochore at the centromere, they are called kinetochoral spindle fiber. So what occur really in the metaphase? This is called as a metaphase. So what occur in the metaphase? In the metaphase, the centriole create the spindle fiber. One is the polar spindle fiber, the second is the estaural, and the third one is the kinetochoral spindle fiber, which attaches with each chromosome at the centromere and the kinetochore of the chromosome, and they arrange the chromosome on a plate-like structure. You can see here, this is a plate-like structure arranged in between the cell in a plate-like structure. So they call as a metaphase plate. This is called metaphase plate. Now the cell will have to enter another phase called anaphase. In the anaphase, so this is a cell. It have to enter the anaphase. So this is the anaphase. Okay. These are the centriole. Okay. Okay, so what occur in the anaphase? In the anaphase, the spindle fiber which were attached at the centromere with each sister chromatid, now the kinetochoral microtubule pull the sister chromatid to the different pole of the cell. Now the cell have to enter another phase called telophase. In the telophase, the completion of the cell division occur. This is a cell. So it is a cell. In this phase, the chromosome reach to different pole of the cell. So there is also chromosome. There is also two chromosome, and there is also two chromosome. and the wall of the cell move inward. Okay. Would occur in the telophase. In the telophase, the chromosome reach to different pole of the cell, and the centriole, the spindle fiber formed by the centriole here, the spindle fiber disappear completely, and the nuclear membrane again appear. This is a nuclear membrane, and this is also a nuclear membrane. So what occur in the telophase? In the telophase, the chromosome reach to different pole of the cell, and the spindle fiber are disappeared, and the nuclear membrane again appear and rape from the nucleic genetic material called chromosome, and the cell membrane move inward. So now the cell have to completely divided. So now this was the division of the Cytokinesis. Now this is there also another type of division which is called the Cytokinesis. And the Cytokinesis is simple short story that when this nuclear membrane, this cell membrane will move inward and completely move inward, and the animal cell, the procleavage appear which move inward, and the creation of two different cells occur. While in plant cell, this wall, like I said, this wall is made of poro or cleavage in the animal cell, while in the plant cell, as we say that Golgi apparatus contain the vessels. So in the plant cell, the vessels come like this, and the vessel appear like this structure, and they completely form the cell membrane and give another cell. So in the plant cell, the Golgi apparatus vessel comes and form the cell membrane, which called then the cell membrane form it, or this called as a cell wall of the plant cell, and two daughter cell are formed. Okay. There are also some checkpoints in this cycle that enable the cell to be move further in the mitosis phase or not. So there, before the space, there are checkpoint. There is a checkpoint which we call as a G1 checkpoint. G1 checkpoint. At this checkpoint, like if we are going some other country, so the police check our visa card, etc. What are our nationality to the country? So when the cell reach to this stage to be undergo the synthesis phase in order to replicate its genetic material, it has also to be checked firstly. Why? Because in the G1 phase, we say that there is the synthesis of the protein and enzyme occur for the duplicating of the genetic material. So this point check that are the protein and enzyme are completely enough for the duplicating of genetic material or not. So this checkpoint is very important. And there is also another checkpoint there. This is called G2 phase, G2 checkpoint. At this checkpoint also checking occur. That was the G1 phase. At this point also the checking of the cell occur would occur when in the S phase we say that the duplicating of the genetic material occur. So at this point the cell also check this, that are the genetic material are completely duplicated or not, are there is some mutation in it, or some like we say that we have 46 chromosome. So if we say that our 10 chromosome or 13 chromosome is not duplicated, so what will occur? So the cell will not move in the mitosis phase and will stay there and come back to the S phase in order to the remaining 10 chromosome to be duplicated, and then will move forward. So in this phase would occur the checking of the chromosome occur that are all the 46 chromosome are duplicated or not. So at this point, this checkpoint is also very important. If this is not, this is not, so there will be some non-duplicating chromosome occur, and then will be the person will be mutant, and something else will be formed, and the cell will be not a normal cell. So this was about the life cycle, cell life cycle. So cell life cycle is consists of interphase and the mitosis. And interphase, the cell prepares itself, and the mitosis phase it undergoes the four phases: prophase, metaphase, anaphase, and telophase, in which it divide its genetic material chromosome and move to different part of the cell, and then the cleavage or furrow on the plant cell, the mitochondrial nut, the Golgi apparatus vessels occur in between and form a chain-like structure and differentiate the cell from each other and form a cell wall or cell membrane in between. So I hope this lecture may be helpful to you, and if you have any doubt, any question in this, so you can mention in the comment, and please subscribe and share my lecture for next video. See you in the next.
Please subscribe, like, and share Ramzan Biology for next lectures.